Okay. The next part is for looking for the target of transcription factor binding. So initially, when first ChIP was invented, it was done in ChIP-seq in yeast. I remember yeast was pretty easy. Each gene is 1 to 2 kb long. From this gene to the next gene is only 1 kb. Therefore, if a transcription factor is binding in that intergenic region, you know it will just regulate the nearby gene. It's fairly simple. However, when we were doing ChIP-seq in human or ChIP-seq in human, one thing we found is if you look at where this transcription factor is binding across the whole human genome, for a lot of transcription factors, you will see this case. The promoter has binding, but that's only 3% of the binding sites that appear within 1 kb from the gene start, transcription start site. So 3% binds to the promoter, but exon, very small, 1% bind to the exon, but 35% bind to the, or let's see, this is kind of intergenic, but still pretty close. Yeah, 35, well, intergenic, less than 25 kb away is 8%. Over 25 kb away from a gene is 35%. And then 48% binding to the introns. And so this creates a big problem on how to really assign target, right? Because you have so many remote controls. Which one is linked to which? It's very, very difficult. So very early days, people just look at a very ad hoc way. They say, okay, we're going to call a target if it has a binding near the promoter. But then you ask, how far away do we call promoter? In yeast, it's like 1 kb. But in human, is it 1 kb? Is it 3 kb? Or 5 kb? Or 10 kb? It's really quite ad hoc. Everybody is trying something different. It's kind of crazy. And you can see actually from homework how well it works. And there are also things that say, okay, for each peak, I'm just going to assign it to the nearest gene. You count the distance from this peak to the transcription start site. Usually, if it appears in the intron, it doesn't necessarily just bind to the gene. For example, let's look at this case. If you have a gene that's really far, and the transcription start site is right here, and they say there is another gene that's going in this direction, and so supposedly these are the exons, right? So these are the exons for this gene, and if you have a transcription factor that binds to this place, it's likely to regulate this gene, probably more likely than to regulate its own gene that it resides in because it's closer to the transcription start site of the nearby for another gene. Okay, so usually there are algorithms that is available to assign this to the nearest gene. But how far do you go? Do you go to 1 meg? It's still kind of also ad hoc. And also, there are people looking at the number of binding. That's actually important. You can find that, say, if there is a gene nearby, there are a lot of binding sites. Okay, this is probably a real target for the transcription factor. So people did find that. And maybe you can use other knowledge. This is still really an open question. And so right now, at least what we use is, you ask, okay, what is the TF's real target? We say, okay, we need to look at different evidence. One is you need to look at binding. So a gene is more likely, a TF, a transcription factor target, if this transcription factor has many binding sites that's fairly close or that are closer to the transcription start site of this gene. However, not all the genes with binding are direct targets in one condition. For example, we mentioned that in order to regulate a gene, the transcription factor needs to have a kind of interaction with another factor of TF2 and the TF3 and TF4. Maybe in this particular condition, you only have TF1. The TF2 and 3 are still waiting. Maybe it's waiting for a heat shock. Maybe it's waiting for a starvation or some immune stimulation that's not there. Then that TF, the first TF, may not have a productive binding. It just binds there and wait, right? So this may not be the real target. And then, so in order to really call it a target, not only we need to have binding, we also need to look at expression evidence. So if this TF is being perturbed, say you activate the TF, or you knock down the TF, or you overexpress the TF, do you see this target gene being differentially expressed? And if that is the case, maybe that's also a target. The question is, how do we combine the binding evidence and the differential expression evidence together? So we actually do it this way. To model the TF binding target, people have found that in general, if you have a gene like this, there could be many binding nearby that they can loop to the, like they form this higher chromatin loop. We'll have one lecture specific for this. There's like looping. And if you look at how much chromatin can loop, you can see there is like this exponential decay. Things that are close by are more likely to interact with each other, but as the chromatin are further and further away, they are less likely to interact with each other. And most of this decay will finish around 100. Actually, this is 100 megabase. That's really too far. But anyway, so it follows this exponential decay. And so what we use is like this. We look at everything within 100 kb, but we use a decay distance. So for example, if we use a decay distance of 10 kb, it would mean that if you have binding right at transcription start site, that's counted as one binding. But if you have a binding that's 10 kb away, that's only counted as half a binding. But if it's a 20 kb away, that's only a quarter of a binding. And like it just every 10 kb it decays, and eventually it will by 100 kb it pretty much has very low effect. And so basically for every gene, we just ask near this gene how many binding do we have, and we weight each binding by their distance with this exponential decay. And based on this, we can rank all the genes based on, we say, okay, this one has a very good binding evidence. The second gene has lower binding evidence. Third gene has, okay binding evidence. And so this way we can create a rank order of all the genes just based on binding. But then we can also rank the genes based on expression. For example, very often people do ChIP-seq in a cell, but they do when the transcription factor is active. But then they also do two different gene expression. When the factor is active, the other one when the factor is inactive. You can also look at their differential expression. Okay, so you look at their differential expression when the transcription factor is activated or repressed, and you ask what genes are differentially expressed. Because those differentially expressed genes are also likely to be the target. Or if you look at the tumors or tissues to ask which genes are correlated with your TF of interest, those are also likely target. And we use either the differential expression level or the correlation level to create a second rank. And at the end, we do a rank product of the two. So then basically, you say, if this gene has a lot of binding nearby, and when you perturb this transcription factor, this gene has differential expression, that's a true direct target of the transcription factor. Does that make sense? Yeah, so you need to have both binding evidence and the differential expression or correlated expression evidence, and you use both to do the ranking. And then at the end, you calculate a rank product to get the target genes. And so in homework 4, we're going to ask you to do this. In fact, we have a tool. You just plug this in on a web server. It calculates this for you automatically. Okay? And what you find is that a transcription factor might be binding to the genome around, you could easily have 20 to 30,000 binding sites. I remember when we were first publishing this, the biggest problem reviewers had is when it's binding to the genome 3,000 locations, how can it regulate a few thousand genes? The fact is, whether it regulates or not regulate is not a binary yes, no. We're saying this is a very tight tightly strong target, and this one is a weaker target. The next one is a weaker target. And so when you combine the gene expression, the binding data and the gene expression data, you can have a rank ordered list of which ones are the strong target, which ones are the weaker target. And so in each condition, it's not surprising for a transcription factor just to have maybe a few hundred strong target, even though they bind to 20,000 locations in the genome. Okay, and so how do we know what are the target gene function? If using this, we get the rank order, we take the top 200 to 300 genes, what do you do to find out their function? Yes. Yes, you can. Yeah, you can run like David. You can run gene set enrichment on those few hundred genes and see whether you see enrichment. We didn't have, so the tool you're going to try is the Systrom Go. You can also try David to see. Systrom Go only use gene ontology. We didn't have a gene set enrichment implementation. But these are some of the functions that can be very useful. It will tell you what's the direct target of this transcription factor, and what's the function, or what are the target function are. Okay. So in summary, ChIP-seq is a very, very powerful experiment. It can identify the genome-wide. In vivo means it's in the cell. It's not just binding to the DNA. Yeah, it can do genome-wide in vivo protein-DNA interaction sites. And you can do, you can use Max for ChIP-seq peak calling. And also for quality control, there's like standard fast QC stuff, but after you run Max, you can get, FDR, you can get the fold change, you can look at conservation, you can look at motifs to look at, to really quality control your sample. By the way, if you look at the genome to look at where this motif appears, because the human genome is a 3 billion base pair, this motif usually appears a lot more times in the genome. Because when your genome is long, the motif will just appear many times. A real transcription factor binding only go to a very small subset of those motif occurrences, you will see. And also go to its collaborating factors motif. But after you call the peaks, there are a lot of downstream things you want to do. Motif finding is very, very useful. You can use it to make sure that you are getting the correct ChIP-seq done. You can also use it to find what's the collaborating transcription factors with your factor of interest based on the motif. You can also use the ChIP-seq peak overlap to help you figure out, oh, these two factors might be working with each other. And sometimes you can see the same transcription factor might activate gene expression, but also downregulate expressions for other genes. And that is mostly because it's collaborating with different factors. It's like, the same guy can do good things when he hung out with good friends, but do bad things when they hung out with bad friends. It's the same thing. You can see the same transcription. The TF1 can activate gene when it interacts with TF2, but it can downregulate gene when it interacts with TF3. Okay, so you will see that, some of this. And for target identification, we recommend looking at binding sites that could be pretty far away, 100 kb away potentially, but weigh the different binding sites by their distance to the transcription start site. And in addition to using this binding evidence, we also use differential expression to also give us evidence, and combine the two evidence to give you the direct target. And then from there, when you run the gene ontology or pathway analysis, you will get better enrichment than just promoter binding or assigning it to the nearest gene. So hopefully you'll see that in homework 4. Okay, so that's good for today.